Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Langerhans cell. This cell was named after a German pathologist, Paul Langerhans, who identified this cell. Scientists will have only name but no fame. After him only another group of cells in the pancreas is also named, that is islets of Langerhans. Now coming to this Langerhans cell, it is a dendritic cell, which is a key, not this key. Key means very important antigen-presenting cell located within the epidermis, mainly in the stratum spinosum layer. Out of all the cells in the epidermis, our Langerhans cells contributes about 2 to 4% with a density of 460 to 1000 cells per mm square. These Langerhans cells are not only present within the skin, they are also present in oral and genital mucosa, within the crypts of the tonsils, thymus, lymph nodes also. Coming to its origin, that is embryology, first it appears in the epidermis around 10 weeks of gestation itself. It belongs to the monocyte, macrophage and histiocyte or dendrocytic series. It is derived from hematopoietic stem cells from myeloid lineage. From hematopoietic stem cells, we will get two lineages. One is myeloid, one is lymphoid. From lymphoid, T lymphocytes, B lymphocytes and natural killer cells will be derived. From myeloid cells, platelets, RBCs, all other cells will be derived. So here Langerhans cells are also derived from myeloid lineage only. It has a dual origin. One is during embryo stage, it is derived from yolk sac. Another one is during fetal stage, it is derived from mainly liver monocytes. We all know that during fetal stage, hematopoietic organs are liver and spleen. Here, liver monocytes will go and settle down in the epidermis. Later, they will produce or change themselves into Langerhans cells. During adult stage, our Langerhans cells have the capacity to self-renew itself locally. If in case any severe inflammation or severe skin injury happens and depletion of Langerhans occurs, then bone marrow monocytes, which will be circulating in our blood, will come and replace the lost Langerhans cells. In some books, it was given directly, it is derived from bone marrow. From this embryology part, we can draw a flowchart and it will be helpful to fill our exam paper, at least half of the page. We will draw whatever we learnt, embryology of Langerhans cells. First, from yolk sac or liver, we will get hematopoietic stem cells, which have two lineages. One is myeloid lineage, another one is lymphoid. Lymphoid lineage we don't want. We will continue our myeloid lineage. From myeloid lineage, we will get monocytes. From monocytes, we will get our dendritic cell, that is Langerhans cell. This Langerhans cell, if present in epidermis, it is called Langerhans cells. After capturing the antigens from epidermis, it will go into lymphatics. If it is present in lymphatics, it is called as Wheeled cell. And then from there it will go to the nearest lymph node to present the antigen to naive T lymphocytes. There within the lymphocytes, within the lymph node, we will call it as interdigitating dendritic cell. So remember, same Langerhans cell have different names according to the location. In skin, it is Langerhans cell. In lymphatics, it is Weyl cell. In lymph node, it is interdigitating dendritic cell. Remember this point. I will be again teaching you. Clear? If you are lucky enough like me, I will have a very big handwriting, so I can fill almost more than half of the page with this flowchart. Filling all 3 pages, all 3 papers for each question is very important. Coming to demonstration of Langerhans cell. We have known from where it is derived. Now we have to demonstrate. With HNE staining on light microscope, These cells will be visible as suprabasal clear cells called as high level clear cells. It is present on the suprabasal layer, not in the basal layer. So it is one level up. So it is higher level compared to melanocytes, which will be present in basal layer. So melanocytes are called low level clear cells with coffee bean nucleus. Because of during processing the tissue, we will take punch biopsy of the skin, a piece of skin, and then we will be processing the tissue. During processing, cytoplasm will shrink and dendrites won't be visible properly. With light microscope and H&E staining, to make them visible or highlight them, we will use special stains like metal impregnation staining with gold chloride, cobalt, osmium iodide and zinc osmium iodide. Remember, this bracket means osmium iodide is separate one, zinc osmium iodide is also a separate one. Without consuming much space, I have kept bracket. Next, enzyme histochemistry, immunohistochemistry. With the invent of this immunohistochemistry, this metal impregnation staining became obsolete. They are not performed now. Mainly we are performing immunohistochemistry. What is enzyme histochemistry, immunohistochemistry? Our cell will have certain enzymes. We will pour some target substrates. So then it will react and change color or glow. Then we can easily identify it as a Langerhans cell. Same way in immunohistochemistry, we will target one antigen, that is a protein, against which we will pour antibody. It will go and bind and produces a glow or change in color, which can be easily appreciated. Now coming to enzymes, ATPase positive, aminopeptidase positive. Coming to immunohistochemistry, it is CD1A positive, which is a most reliable marker, immunohistochemical marker. And CD207, also known as Langerhans positive. This is the most specific marker of Langerhans cell. When compared between CD1A and CD207, 207 is most specific. S100 positive. This S100 is positive even in melanocytes also. So it is very sensitive. It includes Langerhans cells, melanocytes. Sensitive means it includes all without leaving anything. Specific means it only includes what is there. Particularly, Langerhans cells are specific to CD207 Langerhans. S100 is less specific because melanocytes also shows S100 positivity. MHC class 2 antigen, that is HLA-DR positive and CD45 positive. This CD45 positivity indicates it is of hematopoietic origin. All the leukocytes will have this antigen. That's why it is called leukocyte common antigen. MHC means major histocompatibility antigen. Coming to electron microscope, which is the most specific one. All these here are light microscope features. Here in electron microscope, we can see ultrastructural. It is a dendritic cell with coffee bean nucleus. This coffee bean shaped nucleus is the characteristic of Langerhans cells, which will have a ingrown, infolded, indented nucleus. Single nucleus because it is derived from monocyte. Mono means single nucleus. Coffee bean shaped nucleus. And another main characteristic thing is it will have Birbeck granules, which will be rod shaped. Here you can appreciate rod shape or tennis racket shaped. It will have a vesicle at one end or both end. If it is having a vesicle at one end, it will be appearing as a tennis racket shape. Main specific feature for Langerhans cell. These Birbeck granules are derived from this plasma membrane, cell membrane itself, due to invagination of this cell membrane only. How they identified this? We all know it is CD1A positive. When incubated for some time, after some time this Birbeck granules also became CD1A positive. That means this cell membrane only have invaginated inside and became this Birbeck granule. This formation is induced by our Langerhans, that is CD207. It won't have tonofilaments and desmosomes. If we see tonofilaments, that is keratin intermediate filaments, and desmosomes, it will become a keratinocyte. It won't have melanosomes. If we see melanosomes, it will become a melanocyte. So as a cell, it will have all the other organelles like endoplasmic reticulum, Golgi apparatus, lysosomes. Here I didn't draw them particularly, which is needed. I have drawn dendrites, coffee bean nucleus, Birbeck granules. That's it. You have to remember from electron microscopy. Coming to functions, it is a part of skin mononuclear phagocyte system. What is skin mononuclear phagocyte system? It contains Langerhans cells, dermal dendrocytes, and macrophages. All these cells contribute to skin mononuclear phagocyte system. It participates both in innate and adaptive immunity. Using these dendrites, which extends towards the epidermal surface in a uniform manner, Langerhans cells will trap antigens. Using dendrites, which will be extending towards the surface, if any antigen comes and falls on the skin, it will capture it, that is, traps the antigen. Then it will phagocytose it, as it is a part of phagocyte system. Then it will process, and then it moves to present the antigen. It migrates from skin into the lymphatics. I have told you, I will teach you again. Remember this point. I have told you that veiled cell will be present within the lymphatics. So here it is migrating to the lymphatics. Remember, a whale will swim within the water. This whale, our veiled cell will swim within the lymphatics. Why? To reach the nearest lymph node. So within the lymphatics, our Langerhans cell is called a veiled cell. Now it is reaching the nearest lymph node, where it will present the antigen to naive T lymphocytes. While presenting, it will form digitations. That's why we are calling it in lymph node as interdigitating cell. Here you can see interdigitating cell in lymph node. Clear? In epidermis, Langerhans cell. In lymphatics, veiled cell. In lymph node, interdigitating cell. All three are same but different names because of their structural difference within the location and function also. After presenting to T cells can form effector T cells, cytotoxic T cells, and memory T cells. If it forms memory T cells, next time if the same antigen comes, it will react fast. That is our adaptive immunity. That's why it is playing a role in both innate and adaptive immunity. From this function also, we can draw a flowchart. After another thing, it will show a unique motion between the keratinocytes to cover a wide area and to trap the antigen efficiently. It is D-search. This motion is called as D-search. It is not A search, not B search, not C search. It is D search. Using dendrites, it is forming it is making a surveillance. That is, the full form of D search is dendritic surveillance, extension and retraction cycling habitude. This motion is shown within the epidermis, that is between keratinocytes. Now we will draw flowchart for this function. First, if any antigen enters the skin, it will be trapped by dendrites of Langerhans cells. Then it will be trapped, then phagocytosed, then processed, and then it will migrate to lymphatics. Then it will reach the nearest lymph node, where it will be presenting the antigen. What is a Langerhans cell? It is a key antigen presenting cell. Then it will present the antigen to naive T cells, where from here these T cells will get sensitized, activated, and then they will form effector T cells, memory T cells. Effector T cells may be cytotoxic T cells. This much we can cover entire page if you are skilled enough with this single function itself. Next, it maintains a balance between immunity and tolerance. What is tolerance? If there is no tolerance, our immunity will act against ourselves and will form autoimmune diseases. That is tolerance, knowing what is self-antigen, what is foreign antigen. Suppose if a keratinocyte which is adjacent to Langerhans cell died, apoptosed, then the fragments of that keratinocytes will be engulfed by the Langerhans cell. Same like this antigen, which is foreign antigen. Here our apoptotic keratinocyte is self antigen. No need to produce any immunity against it. That is tolerance. After capturing the keratinocyte, which is self antigen, it will same phagocytose process and then it will stimulate T regulatory cells by releasing cytokines like tumor growth factor, beta, interleukin 10. Regulatory cells will regulate the T cells, won't allow them to activate. It will inhibit the activation of T cells because there is no need to activate any immune system against our own self antigen, that is tolerance. Clear? Remember, a regulator will not allow extra voltage to reach TV. Same here, our T regulatory cells won't allow any T cell to act against our own antigen, that is keratinocyte. If it is failed, there is no tolerance, then immunity will develop against own antigen and leads to autoimmune diseases. Now it is not producing any autoimmunity. Same way we are getting, it is helping in immunity also. This is the immunity flow chart. It will release, our Langerhans cells will release cytokines like interleukin 1. If it captures an foreign antigen, then it will stimulate immunity. We have to act against foreign antigen and destroy it. It will release interleukin 1, stimulates, activates, and these interleukins will form chemotaxis, that is attracts T cells towards the site of foreign antigen to destroy it. That is nothing but this flow chart only, immunity flow chart. Clear all the functions? Mononuclear phagocyte system, innate immunity, adaptive immunity, balance between immunity and tolerance, trapping the foreign antigen. It releases cytokines also. Coming to the conditions, few conditions where our Langerhans cells will reduce in number, where it will increase. It will reduce due to chronic repeated UV exposure, elderly. If when we become old, anyway we will be exposing to UV rays continuously, chronically. Application of potent topical corticosteroids and systemic corticosteroids also. Same, this bracket means both systemic corticosteroids as well as topical corticosteroids. Within the warts and psoriatic plaques, Langerhans cells will be depleted. And in HIV, where patient will be immunosuppressed, there will be no Langerhans cells. In HIV, Langerhans cells are among the first cells which will be attacked. HIV virus enters the Langerhans cells. Langerhans cells will think it as a foreign antigen, will go to the lymph node and presents it to CD4 T cells. From there, that virus will spread systemically to all the CD4 T cells. Where Langerhans cells will be increased, eczematous dermatitis like atopic dermatitis, contact dermatitis, drug-induced lichenoid eruption, lichen planus, and Langerhans cell histiocytosis. We all know that in Langerhans cell histiocytosis, there will be clonal proliferation of cells which will be having big granules, same like Langerhans cells. I have covered this topic already and posted in my channel. We will see a few differences between a Langerhans cell and melanocyte. Our next topic is melanocyte. We will have an intro here itself. We all know that Langerhans cells are suprabasal clear cells. Melanocytes are basal. Melanocytes will have a small oval nucleus. Here Langerhans cells have indented nucleus. What is the special stain in Langerhans cell? Metal impregnation staining techniques. Here for melanocytes, Mason Fontana, which will stain melanin granule as black. This is a special stain. Langerhans cell is ATPase positive. Our melanocyte is DOPA positive. This DOPA reaction is shown by tyrosinase enzyme. Coming to immunohistochemistry, we have now only seen that it is CD1A positive, CD2 not 7 positive, S100 positive. Same melanocyte is also S100 positive. Along with that, HMB45 and melan A positive. Both are dendritic cells. Both have a nucleus, but difference is here it is coffee bean shaped, here it is oval shaped, which is a small nucleus. Here characteristic feature is Birbeck granule, here it is melanosomes. Both are not keratinocytes, so obviously both of them won't have tonofilaments, desmosomes. Coming to origin. What is the origin? Embryology of Langerhans cells, it is hematopoietic in origin. Melanosomes are neural crest origin. Upon chronic exposure to UV radiation, what will happen? If we use common sense, melanocytes will increase. Langerhans cells will decrease. Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. До свидания.